Welcome to the latest webinar in Teagasc's Research Insights series. This series is focused on going through the challenges and opportunities in the agri-food sector and in showcasing the latest in Teagasc research. Today's webinar is brought to you from the Food Research Programme, which is based at our research centres in Ashtown in Dublin and Moorpark in Fermoy. And the topic today is viruses. It's almost a year since the first COVID-19 case was reported in Ireland. And today we're going to hear from Dr. Fiona Crispie and Dr. John Kenny, who are part of the Irish Coronavirus Sequencing Consortium, a group of scientists nationwide who are monitoring the virus variants that are currently circulating across Ireland. But despite the negative connotations we all associate with viruses at the moment, there are viruses that can be used to our benefit. Today, Dr. Olivia McAuliffe is going to speak to us about Bacteriophage, which are bacterial viruses that may be used to improve the safety and quality of our food. After the three presentations today, we'll have a Q&A session. If you have any questions for our presenters, please pop them in the Q&A box and include the name of the person the question is addressing. So to begin today, we've got Dr. Fiona Crispie. Fiona is based at Teagasc Moorpark, where she manages the DNA sequencing facility. Her research interests are the microbiome of the human gut, its effect on our metabolic and mental health, and its modulation by diet and exercise. So I'll hand over to you, Fiona, please.
Hello, everybody. My name is Fiona Crispie, and I'm a senior technologist here in Teagasc Moorpark, and I'm here today to give a talk entitled Knock Knock, Who's There? Using sequencing to open the door to the microbial world. So during my talk, I'm going to try and explain a few things. I'm going to try and explain what DNA sequencing is and the information that DNA sequencing gives us. I'm also going to tell you briefly about the DNA sequencing facilities that we have here in Teagasc, and also I'll talk about some of the things that we use DNA sequencing for in Teagasc. So firstly, I'm going to have to try and explain what DNA sequencing is, and for this I'm going to have to tell you a little bit about DNA structure and about the genetic code. So I'm sure all of you know that DNA is a molecule that's found in all our cells, and it basically determines things like whether you have brown eyes or blue eyes, or whether or not you can taste cucumber. It also determines more serious things like whether you have conditions like cystic fibrosis or hemophilia. So DNA is made up of molecules that are called nucleotides, and these nucleotides are made up of a sugar and phosphate group and another group called base, or a nitrogen base. Now, these sugar and phosphate groups aren't very important in terms of DNA sequence, but they join together and form a chain. If you think of a pearl necklace, this chain would be like the string the pearls sit upon. Then protruding out from the chain, you have these nitrogen bases that I referred to, and these would be like the pearls on your string, on the string on your necklace. So these bases, there's 4 of them in total, called thymine, adenine, guanine, and cytosine, or we call them A, T, G, and C for short, and these protrude out from your chain. Then each base will form a bond or glue itself together with a base on another chain. And this actually happens in a very specific way, such that thymine always bonds with adenine, T with A, and G with C. And then the 2 chains, because they've been glued together by these bases, they actually twist around each other, forming this double helix. So another way to view it would be to consider the two long strings of sugar phosphates, or your backbone of your DNA, as the 2 side rungs on a ladder. And then the pairs of bases that are formed in the middle, that are glued together, these would form your foot rungs on your ladder. And then your ladder twists around itself, forming this helical molecule. So what's that got to do with the color of your eyes? So the order of the bases along your strand of DNA is what determines DNA's instructions, or your genetic code. So every 3 base pairs along a strand of DNA codes for something called an amino acid. And amino acids are the building blocks of proteins. So all the proteins that are found in our cells, and indeed in the cells of all living creatures, are made up of amino acids, and these in turn are encoded by DNA. And different combinations of amino acids will give you different proteins. So for people that have blue eyes, you'll get a certain stretch of DNA encoding certain amino acids that then make a protein for blue eyes. In people with brown eyes, the DNA has changed, so the amino acids are changed and the protein is changed, giving them brown eyes. There are only 21 amino acids in total. So in actual fact, some amino acids can be coded for by more than one trio of bases. So for instance, phenylalanine, which is an amino acid that's often used in nutritional supplements, that can be encoded by two different codons, or trio of bases, as we call them codons. It can be encoded by either TTC or TTT. Other amino acids like methionine can only be encoded by one code, like HEG. So this is something I'll probably mention again later on in the talk. And when cells replicate in nature, a protein called a polymerase copies the strands of DNA almost like a photocopier. And with DNA sequencing, we basically just replicate what happens in nature. So we manipulate these enzymes, these polymerases, and we get them to copy the DNA strand over and over again, enabling us then to just read along it and see the order of the bases on the strand. And then this order of the bases will tell us what amino acids are encoded and therefore what proteins are produced. So your entire string, or your pearl necklace of DNA that's found in your cells, that's called your genome. And genomes vary massively in size. So the human genome is very big and very complex. There's actually about 8 billion base pairs in it. But microorganisms have much smaller genomes. So for instance, the bacteria that you'd sometimes find in yogurts, probiotic bacteria, they often only range from about 2 to 3 million base pairs in size. And then other viruses or other genomes, for instance, the genomes of viruses are smaller again. So for instance, SARS-CoV-2, which is the virus which encodes COVID-19, actually only has about 30,000 bases. It's a very, very small genome. And it's actually fascinating to think that something so simple can actually cause so much damage. But in actual fact, it's almost elegant in its simplicity. This virus really only wants to replicate. It encodes very few proteins, and most of them are involved in viral replication. And its small genome size is also reflected in its small actual size. Viruses are amongst the smallest living organisms known to man. And it has recently been estimated that if you could gather all the COVID viral particles that are in the world right now, you could actually fit them into 1 to 2 cans of Coke. It's that small. So just as I said a minute ago, the genomes of humans and indeed of animals as well are far bigger and more complex than those of microorganisms. So if you were to view your genetic code as, or your genome, sorry, as a book, the human genome would probably be a book like Ulysses, very long and very complicated and difficult to follow at times. Whereas the genome of microorganisms, in particular of viruses, because it's so much more simple and shorter, it would be like a children's book. So for instance, The Cat in the Hat. And then to take that analogy a little bit further, you could consider the 30,000 bases that's encoded, say, in the SARS-CoV-2 viral genome. They would be like all the letters in the book. And then these letters form words, or your amino acids. Those words form sentences, and they're the genes that are encoded. And those sentences then encode, or those sentences then make paragraphs, or the proteins that are encoded by the virus. And then for SARS-CoV-2, for instance, 4 proteins are on the surface of that virus, and that would be like your key chapter in your book, the most important chapter. So when viruses, or when any DNA is replicated in nature, sometimes the photocopying enzymes make mistakes, and this is called a mutation. And viruses with different mutations are referred to as variants or lineages, and you've probably heard a lot about SARS-CoV-2 variants on the news lately. To go back to our children's book analogy, mutations are like a typo in the text. So sometimes this is very subtle and it doesn't really matter too much. So for instance, if you look at this sentence here, the cat is in the room. This is a sentence you'd find in your nice simple children's book. And the first type of mutation would be where maybe one letter of a word is changed, but the amino acid isn't changed at all. So this was, if you remember earlier, I said phenylalanine could be encoded by 2 different codons. This would be a case where one of those codons was switched for another. So in actual fact, the amino acid doesn't change and the protein doesn't change. So your sentences change very subtly, but it's no big deal. That's what we call a silent mutation, or a synonymous mutation. A second type of mutation is where you do change the amino acid. So for instance, if we switch the C of cat for N, now we have a different sentence completely. The mat is in the room. So in this case, the sentence is messed up, but again, it's not really anything to worry about massively. In fact, it probably makes our whole story a little bit more boring. It's not that interesting. And in this case, that would be where an amino acid is switched for another one that makes your protein less harmful. However, a third type of mutation can occur. So this would be something like where you switch the C of cat for R. Now, all of a sudden, we have a sentence that reads, the rat is in the room. So suddenly your key paragraph becomes far more sinister and threatening. And this is an example where an amino acid in a protein is changed in such a way that it makes the protein more harmful. So the mutations that occur, say, for instance, in SARS-CoV-2 that make it more able to bind to our cells more efficiently, or that make it evade the immune system, are just simple mutations like this, where one letter in the genetic code is changed, but that meant a different amino acid was produced and thus a different protein, making the virus more harmful in some cases. So differences in DNA sequences can also be used to distinguish other organisms from each other. It's not just used to distinguish viral variants. So for instance, humans share 99% of our DNA with chimpanzees. Now, I don't think we really need to do DNA sequencing to be able to tell the difference between us and chimpanzees, but it is useful to do it just to show how we've evolved. What we can use it for, though, is to distinguish between different bacteria as well as different viruses. And again, this is something I'm going to come back to later on in the talk. So next, I'm just going to mention briefly the DNA facilities that we have here in Teagasc, and in particular in Teagasc Moorpark. In Teagasc Moorpark, we have a fairly large DNA sequencing facility, and it contains all of these DNA sequencers that you can see here. I'm not going to go into detail about all of these except to highlight two, the MinINE here and the GridINE here. So the MinINE is a handheld sequencing device from Oxford Nanopore Technology. And the GridINE is like the big brother of the MinINE. And both of these sequencers, I'm going to mention the MinINE again later on in my talk. And I think John is going to mention both of them during his talk. So what's all of this got to do with sequencing, or with Teagasc? You might be surprised that a Food and Agricultural Institute would be so involved in DNA sequencing. Well, actually, we apply DNA sequencing in lots of different ways. So one of the main areas that we've been applying DNA sequencing for is in microbiome research. So in recent years, a lot of research has focused on our microbiome. So our microbiome is the collection of microorganisms that live in and on our bodies. And some of you may know this already, but some may be surprised to hear that in actual fact, the human body contains trillions of microbial cells. And in fact, the microbes in and on our bodies outnumber our human cells by about 10 to 1. And collectively, the genomes of those microbes, even though individually they're all quite simple, collectively, their genome content gives us about 100 to 150 times more genes than our complex human genome. So these microbes are now believed to contribute to our health and well-being in numerous ways. So for instance, they play a role in our immunity, in our digestion of food, in regulation of vitamins and hormones, and can even affect our behavior. In fact, they affect our health so much that they're now being referred to as the forgotten organ. And until recently, we found it quite difficult to identify all the microbes living in and on our bodies because a lot of them have complex nutritional requirements, so it made it really difficult to try and grow them in the lab the way we would traditionally grow microorganisms. So the advances, however, in DNA sequencing has meant that we can now sequence all the microbial DNA that we have in a given sample. And then using those differences in the DNA that I referred to earlier, we can distinguish between different bacterial strains and also between different viruses. And it enables us to identify all the good bacteria and viruses in our sample. So in Teagasc, together with our partners in APC Ireland, we do this to determine the effects of dietary and other interventions on our microbiome. So for instance, we've looked at things like the effects of probiotics and prebiotics. And we also look to see if we can improve our overall health and well-being through the food that we eat. Another area that we've used DNA sequencing for is looking more closely at fermented foods. So there's quite a number of putatively health promoting food and beverages on the market at the moment, but unfortunately, a lot of the time, the science behind these can be quite poor. So we're trying to examine this by doing a more rigorous examination of the microorganisms present and the health benefits that they may confer. And by selecting the microorganisms, the good microorganisms from this food, we also hope to generate fermented foods and beverages that closely resemble the artisanal products, but for which the health claims have been proven. And finally, by having these products with the microorganisms we've selected, we'd hope to make the products more flavoursome and improve the quality, and do that more on a more consistent basis than really happens with the artisanal products. A third area that we use DNA sequencing for is looking at animal health and things like methane emissions. So Dr. Matt McCabe in Teagasc Grange has done a lot of work in this regard, looking at manipulating the rumen microflora in beef cattle to see if you can reduce methane emissions. And we've done some similar work on a smaller scale here in Moorpark as well with the dairy herd. And more recently, Matt has been involved in looking at trying to identify bovine respiratory viruses. And actually to do this, Matt is using the MinION, that handheld sequencer that I mentioned earlier. So basically, Matt goes out into the field, takes swab samples from cattle in the field, and then uses a rapid preparation system to extract the microbial DNA and prepare the sample for sequencing. And here you can see he's loading his sample onto the flow cell of the MinION in his portable laboratory, which is also known as his car. And we also have another member of his team then analysing the results on her laptop in the car. So this is really interesting work, as rapid and accurate identification of respiratory pathogens in a herd could mean that, first of all, the therapeutics can be more targeted, but secondly, it also helps us to identify the pathogens correctly, enabling us to develop better therapeutics and better vaccines. So a final area in which DNA sequencing is used is that of food safety and quality. So for instance, identifying microbes that are responsible for food defects or food poisoning, or exploiting bacteria and viruses for food safety purposes. And Olivia is going to talk a little bit about this during her talk. So in summary, DNA sequencing is used for a vast array of things in Teagasc. So on the food side, it's for looking at the composition of foods like cheese and yogurts, including artisanal products, looking at the composition of health promoting foods and beverages, identifying microbes responsible for food defects, exploiting microbes for food safety purposes and to produce nicer tasting foods. And we also have used sequencing for human and animal microbiome studies, where we look at the effects of things like probiotics, antipsychotics, and prebiotics on our microbiome. We look at interventions like diet and exercise. We've looked at ways of manipulating microbes to reduce methane emissions, and we've used it to identify animal pathogens. So as you can see here in Moorpark, and indeed in the other Teagasc centres, we have a lot of experience in DNA sequencing of microbes and have the capability to do so. And this meant when the pandemic began, we were quickly approached by hospital consultants with a view to sequencing COVID variants. And Teagasc was really ideally placed to support the national effort in this regard, and particularly with the expertise and the equipment that we had here at the DNA sequencing facility. So this ultimately led to the creation of the Irish Coronavirus Sequencing Consortium, and some of whom are pictured here in one of our Zoom calls. And John is going to talk to you about the consortium and the work we've been doing during his talk. So finally, I can't finish without thanking some people. All of the work here, and indeed the sequencing platform itself, is not just supported by Teagasc, but also by APC Microbiome Ireland and by science and by funding from Science Foundation Ireland. The work that I've mentioned during the talk is done by a huge host of researchers, some of whom are pictured here, but I'd like to pay particular credit to the other members of the sequencing team, particularly Elaine Lawton, Callum Walsh, and Laura Finnegan. And Amy Fitzpatrick also deserves a special mention for her contribution to the COVID sequencing project. So that's it. Thanks very much.
Thanks, Fiona. That was a really nice overview of DNA sequencing. So as Fiona said, next up is John Kenny, and he's going to continue along the same theme of the Irish Coronavirus Sequencing Consortium. John is a senior researcher in Moorpark, and aside from his work with the consortium, his research interests lie in using new methods to track the microbial composition of environmental niches and how this can then be used for biotransformation and health. So over to you, John. Thank you.
So good morning, everybody, and thanks for the introduction, Muireann and Fiona. So yes, I'm going to give a quick overview of the Irish Coronavirus Sequencing Consortium, and I'll give you an idea of the what, the where, the when, the how of what the project entails. And to start with, it's kind of the who. So Fiona mentioned it. The members of the consortium, the scientists involved are drawn from across the country. And the nature of the project is slightly different to a standard sequencing project. So in most cases, what we would do is we come up with a plan for our project, we write a grant application to get the funding in place to do the work. And then the intention at the end is that we'll publish our data, we'll produce scientific papers, maybe we'll have a product or some sort of translatable approach afterwards. The project here is a little bit different. The reason being that we're going to make the data publicly available as we go along. We're going to do that on publicly accessible websites, particularly a website called GizAid. And we're going to make the sequences available because we want other scientists from around the world to access that sequencing data as quickly as they can in a global effort against the virus. And it's probably important to state here that a lot of the samples we're handling, it's different from some of the work that the National Virus Reference Laboratory, the NVRL, is working on. So a lot of the samples we're looking at are more maybe hospitalized patients and stuff like that, rather than some of the samples that are circulating in the community. So what are we looking at? Well, we're looking at the SARS-CoV-2 genome, as Fiona mentioned. That's the causative virus for COVID. And she said that the genome is about those 30,000 bases in length. And this infographic here, it gives you an idea of the arrangement of the genes across that genome, and that these genes code for proteins. And when they're in the cell and you get the virus infection, what happens is all these proteins combine to generate your final virus. Now, why sequence the genome? What's the purpose of it? Well, we know mutations are going to happen all the time. It's a completely natural component of biology. It's especially expected when a virus jumps from an original host. In the case of SARS-CoV-2, the original host was probably bats. And it's jumped from infecting bats to infecting humans. And that means that it encounters a new environment. You know, our structure of our lungs is different, our structure of our bodies is different, and our immune systems are different. So you're going to expect mutations of the virus to arise naturally, and you're going to expect those variants, those mutated versions of the virus to develop. And back in March, when we first put this application together and we submitted it to Science Foundation Ireland, and they gave us the funds to do the work, we thought that this would be interesting to be able to track those variants, track those mutants in Ireland, particularly with the view to things like vaccine development. So in terms of the vaccine, most of the vaccines that are being developed on the market are directed at this spike protein. And that spike protein, it's on the surface of the virus, it's what your immune system sees. But importantly, that's the kind of structure that makes contact with your cells. So it's like a lock, it's like the key that unlocks the cells and gains access for the virus into your cells to start the infection process. So how are we doing it? Well, if a patient goes for their vaccine, their COVID test, their PCR test, normally what happens is the healthcare worker will take a swab and they'll put that down the back of the throat, the nose. They'll try and get some of the virus from the patient's system that way. And they take that swab, that Q-tip, and they put it into a lysis buffer. And really all that buffer does is it breaks open the virus. It inactivates the virus and it releases the genome. They clean that up, they get rid of the proteins and fats and stuff, so they're left with the virus genome. And then they convert that, they carry through to the PCR detection. So what they're looking to see in the diagnostics test is the virus genome present. Now what we do is ever so slightly different. We take this bit here, so there's leftover material at this stage because not all of the material is needed for the diagnostic test. So we take that leftover RNA and we use that for our sequencing. And how do we do that? What's the method by which we do that sequencing? Well, the viral genome is about 30,000 bases in length, but it's difficult to sequence those 30,000 bases in one go for technical reasons. So what we do is we kind of split the virus up into small chunks of about 400 bases, and we sequence those small chunks, and then we stick them back together using computational methods. And really, it's kind of like a big jigsaw, right? So we've taken the full picture, we split it into small bits, and then we stick it back together afterwards. And when we stick it back together, it gives us the mutation profile of the virus from that swab patient. So it gives us the variant profile for that sample. And how do we do the analysis? One of the ways of looking at it is we want to look at the variants. We want to look at the sequences of the virus that are closely related or the ones that are different from each other. And one of the ways we visualize this, we do this, is to look at something like a genetic tree like this. So, you know, conceptually, we just kind of know that small dogs, kind of terriers, they're just likely to be more closely related. Their genomes are more likely to be similar to each other than they are to larger dogs or wolves, right? So what this genetic tree is telling us is that the genomes of these terriers, they're closely together, they're clustered together on the genetic tree, and that indicates that they're close together. Whereas other dogs, more different dogs to the terriers, they're further away on the tree. And we can apply those we can use the same kind of approach to look at viral sequences. So this is the same kind of genomic tree. We see each one of these points along here is a viral genome that's been sequenced. The ones that are closer together are similar to each other. And these virus genomes, these have been taken from genomes isolated all around the world. So Africa, America, Asia, and so on. Now, the ones that are circled here, these are Irish genomes, right? These are genomes of virus that were taken from patients in Ireland before June the 25th of last year. And what we see is that these are clustered. They're not really in one cluster together, but instead they're all around this tree. And that tells us that we had multiple independent introductions of the virus into Ireland during the first wave of the pandemic. It wasn't just one single introduction. And what you can do with those variants as well is it allows you to look at when those variants arise and look at them over time. So I'm going to quit out of this just for a second, and hopefully you can see this this website here. So this graphic is taken from the GizAid website, and that's where we upload our sequence information. Over on the right-hand side here, we have an image which is the genetic tree again. It's arrayed, instead of being in a circle, it's kind of arrayed horizontally. There's no real difference. That could be generated as a circle. But the thing here I want you to look at is the fact that there's a time scale down the bottom. So what I'm going to press play on this automation. And what we'll see is you'll see a little kind of a white bar appear, and that will start moving towards the right. And the genomes, each one of these dots represents a genome that we've sequenced. And those genomes are going to start appearing on the Irish map here. Now, because we're mapping the presence of the variants against time, and because we know those variants have been detected in other countries, it allows you to kind of track the transmission of the virus into Ireland, around Ireland, and perhaps back out again. So what you're going to see over here is you're going to see some lines which indicate that the virus has appeared from other countries, and we'll be able to see some lines connecting those between different sites in the country, different cities in the country. So again, over here, we'll see the white bar moving along rightwards to indicate the move through time. And we're going to see these little spots appear, and those spots correspond to particular variants of the virus that we've detected and see. And you're going to see some of these lines appear over here. That's an indication of transmission into Ireland from other countries. And as the pandemic progresses, you'll see some of these lines appearing here, which shows transmission of the virus within Ireland as well. So it's quite a useful approach in some cases for epidemiological purposes. I'm just going to go back to my slides again. And really, Phylogenetics was the title on the slide. That's just a overly complicated, fancy way of looking at those variants against time. Now, what do the variants actually mean? So the image here is taken from New York Times. This is a representation of the spike protein. Earlier on, I had an image, a kind of a graphic of the viral genome, and in that it was 30,000 bases were laid out as if they were kind of linear. Now, in reality, the virus interacts with our cells in 3D. So this is kind of a graphic of the spike protein. The spike protein, how it appears, it's actually 3 versions of the protein that are interact together, that form together. And these bind the receptor on the surface of our cells. And what they're showing here is they're showing specific mutations that you'd see in the spike protein of a virus, a UK variant version of the virus. So for example, we've got an alteration here, and this is the region of the spike protein that binds the surface of the cell. So the alteration here is likely to help those viruses get into the human cell. And we have other variants, other mutations, sorry, on this particular variant at different locations of the protein. And these changes will help the virus likely to fold maybe slightly differently, to evade the immune system a little bit better, and generally do its job when it's trying to infect us as the host a little bit better. So it's important to remember that the mutations take place all the time. It's a totally normal biological process. And when the virus is moved from infecting bats to infecting us, your body applies what we call a selection pressure, right? And what that really means is it starts selecting for viruses which are better at infecting us, which are better at transmitting between one person who's got the virus to another person who doesn't have it yet. So you see that selection event taking place, because when a virus infects a cell, it replicates, it generates more copies of itself. Maybe 1 or 2 of those versions might have a mutation, so that the viruses that get released, you maybe have a mutant version of the virus that gets released. And in most cases, it doesn't matter. That particular virus isn't any better. It's not selected positively. But in some cases, you do get variants, like in the case of the UK variant, where you have a combination of mutations. That means that the virus is just better at transmitting. And really, it's evolution in action. It's kind of what you'd expect to see. So I'm nearly at the end of my section of my talk. And I'm just going to touch a little bit on a publication that we had from the group recently. Excuse me. And these are samples from the All-Ireland Infectious Disease Study. So the All-Ireland Infectious Disease Study is it's headed by Paddy Mallon out of UCD, and really that allows us to take patients for our samples from patients who are hospitalized in Dublin and the southeast region around Wexford. And what it allows us to do is analyze those genomes, but also because Paddy's group have the patient information, that kind of overarching metadata about those patients, it allows us to infer a little bit more from the sequencing information. So this graph is a little bit busy. Bear with me for a few moments. So if we look at the axis around here, this is really just different variants of the virus. So B.11, it's just a label for the particular variant of the virus. And the axis along here is the month, the time at which those samples were collected. So this set of data runs from March of 2020 to December 2020. Over here, these samples in blue, they really correspond with the first wave, which would have been kind of March, April, May of last year. And the samples over here, these were collected towards the second wave, so September, October, November time. And really, there are a couple of things that we want you take note of here. So if we look at these particular variants that are detected here, you'll notice that they're over here, they're in this second wave around this time of the year. And these variants weren't detected at this time of year. They weren't detected at that initial first wave. And what that tells us, it tells us two things that we can infer. One, so these variants were observed that were seen in the first and the second wave. They're different variants. It also suggests what we know about where those variants might have been detected first in other countries. It also suggests that the second wave of infections resulting in hospitalizations, they were largely seeded, they largely came from multiple travel-related events from countries outside the island of Ireland. So if you remember back in kind of the summer of last year, we had relatively low levels. And when we got the second wave here, this second wave wasn't being caused by variants from the first wave, it was being caused by variants from new introductions onto the island of Ireland. So really that's it for me. Just to summarize, for the sequencing side of things, we take surplus sample that's been collected for diagnostic testing. We take some of that material and we do the sequencing of the genomes. As Fiona mentioned earlier on, we do that through the nanopore sequencing. I kind of touched on how we do that data generation and the analysis with the genomic trees. We see that new variants introduced into Ireland after the summer are likely seeded the subsequent infections in the second wave and were detected in hospital patients. And I'm just going to point you at the Teagasc website. If you do want some more details on the project itself, if you go to the Teagasc site and search for the Coronavirus Sequencing Consortium, there you'll find a page with some information there. And that's it for me really for now. And hopefully we have some questions to talk about. Thank you very much.
Thank you, John. That was brilliant. So next up is Dr. Olivia McAuliffe, who's going to speak to us today about bacteriophage. Olivia is a senior researcher at Moorpark, and her research interests are in the genomics of microbiology of food, including starter cultures and food fermentation, as well as bacteriophage in food quality and safety. So thank you, Olivia.
Thanks, Muireann. Thank you very much. And thank you for that introduction. Good morning, everybody. As Muireann said, I'm a principal scientist at Teagasc Food Research Centre, and we're going to change tack a little bit now and maybe stop talking about viruses as something that we need to worry about. And of course, we're all quite worried about the viral situation at the moment. But what we want to talk about is bacterial viruses and their potential role for good. So I'm going to talk about bacterial viruses, or bacteriophages as we call them, or phages for short. So these are viruses that infect bacterial cells only, and they're found everywhere. They're the natural viral predators of bacteria and found in every ecosystem on the planet, anywhere that bacteria are found. So basically in the oceans, in fresh water, in soil, on plant material, and in the human and animal body. And it is estimated that there are about 10 to the 30 bacterial cells on the planet. And for every bacterial cell, there are 10 bacteriophages. So an estimated 10 to the 31, huge number of bacterial viruses on the planet, making them the most abundant biological entity on Earth. And what's different about these viruses compared to SARS-CoV-2 or any of the other viruses that we talk about in general terms is that they infect bacteria only, and they are completely harmless to us, completely harmless to humans and animals. We interact with them every day and they do us no harm whatsoever. And in the graphic here, you can see there's lots of different types of these bacterial viruses, all shapes and sizes. But I guess the most common form that we see are these viruses that look like the lunar docking station docking on the moon. So you've got this head here, which contains the genetic material, a tail, and often coming out of the tail, you've got lots of tail fibers and other accessory proteins that interact with the cell surface. And so here in this image here, you can see some viruses, some bacterial viruses that we isolated in the lab from a sewage sample. And I'm going to come back to source in a minute. but you can see that in the environment, you can identify lots of different types of bacterial viruses, all the different shapes and sizes, some with, smaller heads and very small tails, some with much longer tails and all these tail fibers coming out here. So lots of diversity out there. So how do bacteriophages infect bacterial cells? And I'm going to run this little movie here. So what you can see is the phage docking onto the bacterial cell, injecting its genome, DNA or RNA, into the bacterial cell, replicating within the cell, and then using the host machinery to do that, producing more viruses, and then bursting the bacterial cell open to release those viruses, which then go on and infect other bacterial cells. And that process happens over and over again. And that is how infection and replication of the bacterial viruses happens. And so at Teagasc, we've been studying bacteriophages for quite some time. And our initial work on bacteriophages was because they were problematic, and they were problematic in the dairy industry. And the reason that they were problematic in the dairy industry is because in the dairy industry, we grow large volumes of bacteria. So as I mentioned earlier, when you grow large volumes of bacteria, or if there are bacteria there, viruses will thrive. So we grow large volumes of bacteria if we want to produce fermented dairy products like cheese and yogurt. So in the vat, when you introduce the bacterial culture into the milk, the numbers start to increase, the bacterial numbers start to increase. And if there are phage present and they can get into the factory through the raw milk, in the environment, through personnel, and get into the vat, they can start to cause some severe problems. And this is indicated here on this graph. So if you look at this line here, which is the standard fermentation, so that's when the bacterial cultures start to grow, they produce acid in the vat, and that lowers the pH of the milk, and that's what forms the coagulation. When phage get in, this whole process can slow down because the bacteriophage are killing the bacterial cells, and therefore the bacterial cells are not available to produce that acid and reduce the pH. And this can cause serious economic loss for the dairy industry. And so when I first arrived in Teagasc, which wasn't today or yesterday, this is the type of work that we were doing. But we were also beginning to become interested at that time in phages as biotherapeutics. And this wasn't a new idea, in fact, it was a centuries-old idea. And this looks like a very busy chart, but basically what I want you to see here is this kind of beige curve here, where this is the time point here where bacteriophages were first identified as something that was killing bacterial cells. And you can see that over time, interest in using these agents as biotherapeutics increased. And there was lots of activity all around the world, particularly in the former Soviet Union, in using these bacterial viruses as agents to kill pathogenic bacteria. And this continued on into the 30s and into the early 40s. And then you can see here that the whole thing fell off a cliff. Well, why did the whole thing fall off a cliff? Because Alexander Fleming identified penicillin, and we began to mass produce antibiotics. And so the antibiotic activity and research and commercial activity in this space started to explode. And interest in phage therapy just kind of disappeared in the West, really. In Eastern Europe, they continued to look at it, but not in any major commercial way. And so why are we interested in these things again? Well, antimicrobial resistance is a huge global challenge, and we all know about that. And we need to be looking at alternatives, and phage are one such alternative. And I'm going to try and show you in this presentation what phage can do and in what kind of applications that we can use them. So for control of pathogenic bacteria in the food system, we use two different approaches, and I'm going to talk you through them. Okay, so the first approach that we use is using whole phages. So that's the whole live virus here, either on their own or with other whole phages, and we call these phage cocktails. And we use those to target certain bacterium. And the bacterium I'm going to talk about this morning that we targeted with this approach is an organism called E. coli 0157, which causes a foodborne disease. And I'll come back to that in a second. The second approach that we use is not the whole virus itself, but a part of that virus, an enzyme from that virus. And it's the enzyme that causes that bursting of the cell that I showed you in the cartoon earlier. And those enzymes are called endolysins. And you can use those enzymes not to get lysis from inside to outside, but to get lysis or killing from outside to inside. And the organism that we targeted in one particular study was Listeria monocytogenes, another foodborne pathogen. And again, I'll come back to that in a second. So E. coli first, E. coli 0157. This is an organism that causes a serious foodborne disease. So cattle are the main reservoir of this particular organism, and they shed it in their feces, sometimes very erratically, sometimes quite profoundly. But the cattle are asymptomatic. So what happens is the fecal material gets onto the carcass of the animal during slaughter and gets into the food chain in that way. And therefore, its beef products mainly are problematic when it comes to E. coli 0157. So undercooked beef, minced meat, burgers and things like that. But the food sources are expanding, and people in the food industry are quite concerned about that. It's moving into other food types that wouldn't have normally been associated with this organism. The other worrying thing about it is that there's a high percentage of antibiotic resistance associated with the isolates in animals. So if you become infected with this organism, you know, there may be difficulty in treating that infection because of antimicrobial resistance. So if you're going to target an organism like E. coli with bacteriophage, first of all, you have to go and find the phage. So where do you find them? So as I mentioned at the start, bacteriophage are everywhere bacteria are. So you go back to the source of the bacteria. And if the source of the bacteria is fecal matter of the cows, the slurry is a good place to start to look. So we took bovine slurry samples. You then mix them with your E. coli target cells. You look for lysis or killing. The culture will clear. Normally it would be cloudy. The culture would clear. And then you plate them out. And when you see these little, what we call plaques on a plate of E. coli cells growing, that's where we'll find the phages. And indeed, we did find two particular phages active against E. coli 0157. You can see, you know, I talked a little bit about the shape at the start. These are two different phages. But what was important about these phages was that, sorry, I'm just going back there, is that they were active against all the E. coli 0157 strains that we tested. And that's important because phages can be very specific down to a single strain. And so it was important here that we had some phages that would kill multiple strains, because you don't know what type of infection and which strain is going to be causing the infection that you have. So it was important that we had kind of a broad host range phages here. And just before I talk about the studies that we did, I just want to mention that there's two types of intervention with a phage. So the first one is pre-harvest intervention, where you put the phage on the animal or plant before the animal is slaughtered or the plant is harvested. And then post-harvest intervention, where you put the phage or phage product on either directly onto the food after processing or to the processing environment itself. And so with E. coli 0157, we took a number of different approaches. The first one was a pre-harvest approach. So before the cattle were slaughtered, we put it into the drinking water. This was a kind of a mixed bag really of a result, because what was happening is, as I mentioned earlier, erratic shedding. So the cattle were shedding the E. coli before we could really get a handle on what was happening. But what we did notice, which was very interesting for us, is that when the cattle took the phage in, they excreted it out the other end. So the phage were coming all the way through the animal, which means that in a situation where the organisms weren't being erratically shed, we would potentially be able to control E. coli in that situation. We came up with another strategy, and this strategy was putting the phage on the cattle hide. So I mentioned that if it gets onto the cattle hide, it can get onto the meat during processing. And so this was our approach in this case. And what you can see is there is a reduction in the amount of E. coli on the hide of the cattle when you apply a phage cocktail to the hide. So it does reduce the numbers. In a post-harvest intervention study, we put the phage directly onto the food product. So here we're looking at in the processing plant, when the meat is ready, you put a phage cocktail onto the product itself, like you would maybe any controlled measure. And what we found in that instance is that there were no E. coli detected on 7 out of 9 pieces of steak meat in that scenario. So depending on the application, the phage works. And it's really important to think about the application and the mode of application of these agents each time you consider where you're going to use them. So just coming back to the second approach that we use, and that is using the enzymes or the endolysins, as I mentioned earlier. And this time we targeted an organism called Listeria monocytogenes. This is an organism, again, a foodborne pathogen that causes a disease called listeriosis. And this is a relatively rare infection, which is actually quite good news because it has quite a high fatality rate, over 20% actually. It's primarily associated with ready-to-eat food. So these foods that we buy at the supermarket and take home, deli meats, coleslaw, salads, we put them into the fridge and we eat them directly out of the fridge. So there's no cooking step. And there was quite a large outbreak in 2018, 2019 in South Africa associated with polony, which is like a processed meat, a type of a bologna. And there were over 200 fatalities associated with that outbreak. It was the largest global outbreak of listeriosis. So this is something that we need to be targeting. And so in this case, we had a phage, we identified a phage against Listeria monocytogenes. We sequenced the genome of that phage. So Fiona gave us a very nice presentation earlier on genome sequencing. So we sequenced the genome of this phage and identified the enzyme of interest within the genome. We purified that enzyme. And then we applied that enzyme on the stainless steel surfaces in a food production facility. So here you would see Listeria growing in what's called a biofilm. Listeria love these damp corners of food processing plants. But when you put the enzyme down first, the Listeria can't get a foothold. So if you apply it in advance, the Listeria don't tend to form biofilm. If you put it down after, the enzyme has more difficulty in getting into the biofilm and killing the Listeria. So again, application, very important here, how you do it and when you do it. And it's not just us working in this field. There are lots of commercial activities out there, lots of products which are targeting different bacteria, Listeria again, E. coli, Salmonella, and also some plant-based pathogens. So there's lots of activity in this space. But what is important to note is that there's no common position at the moment on the use of these agents in agri-food systems. In the EU, they're considered as food additives and therefore must be put on the label. And so companies are tending to stay away from that. In the US, they're approved for use as processing aids. So like anything else that you might use in your plant, you can use these agents and you don't have to put it on the label. So we have some work to do in convincing the scientific community and the regulatory community that these are agents that we need to consider. And I really believe that progress in this area is likely to be dictated by public health needs and tackling antimicrobial resistance in the population. And so with that, I'd like to thank all of my colleagues who have helped us in this work over the years, and of course our funding bodies, and to thank you for your attention and take any questions if you have them. Thanks very much indeed.
Thanks very much, Olivia. So I'd ask all our researchers now to turn on your camera and microphones again. We have loads of questions, so I'm going to let the Q&A run on slightly past the 10:30. For those of you asking, the slides will be made available as PDF on the Teagasc website, and the webinar is also being recorded. So you can view that on YouTube on the next day or two. I think Fiona's DNA metaphors have been particularly popular, and I think people would like to see them again. So we have a lot of questions. So I'm going to start off with John and Fiona on the coronavirus. I think the main question we kind of all want to ask, I guess, is when you sequence these new variants, how does that affect the vaccine? And can you tell from the sequence of each variant whether or not it will affect the vaccine efficacy?
You can't really tell from the sequence. You can get some idea of how much the main mutations we worry about would be mutations in the spike protein, because that's the protein that enables the virus to attach to our cells. So we can tell how much the spike protein has changed, and obviously the more it's changed, the more serious it can be. But really you would have to do assays where you would look at the reaction of that virus to antibodies to see whether it would still be efficacious or not. That was why even after the UK variant was detected, it took, you know, quite some time before, well, not a huge amount of time, but a little bit of time before they could say whether the current vaccines would still be effective against it or not. Yeah, does that answer the question, or would you?
Yeah, thanks, Fiona. And one look for John, based on the data you presented, is there anything you can say yet about the third wave, and how is it seeded from the same wave as wave 2 that you presented, or
yeah? So that's a really good question. It's quite a tricky. So I can't give a straight answer. Is the first bit, partly because the levels didn't drop. The levels of community transmission didn't drop to what they looked like during the summer here. So there was a level of transmission that was taking place into kind of the December period that we didn't have during the summer. And what we do see in some of the samples we've sequenced, let's say that the UK variant that was detected in a couple of hospitalized patient samples from end of December. So some of the seeding must have taken place prior to that. Also, because we have these let's say the UK variant, or you know, like some South African variant, Brazilian variant, they look like they transmit better, right? So they transfer easier from me if I've got it to somebody else. And that changes the dynamics of the virus spread as well. So what we're seeing now is ever so slightly different in terms of how the virus is behaving and what it does to the R number compared to previously. Yeah, so it's a slightly different scenario.
Okay, and I've just got one more I'm going to ask John now. Coming from the People's Republic, obviously, can you say which was the most dominant strain in Cork, or can it be narrowed down that far?
I don't think there was any one that was, you know, certainly not those kind of variants of concern. There were none that were massively overrepresented compared to other. And the other thing to remember as well is that the sequencing is useful. It's a useful tool, but we're sequencing a relatively small sample size of all of the variants that are out there, right? So you have to be a little bit cautious about inferring whether we've seen all of the variants that are circulating.
Okay. For Olivia, is there a chance in the future that phage therapy could replace antibiotic therapy in humans?
Yeah, absolutely. And there's lots of work going on in that space as well. And in the past, we've actually worked on that as well, you know, trying to target MRSA with bacteriophages and the enzymes from the phages as well. I guess there was a lot, maybe about 10, 15 years ago, there was an awful lot of funding going into this work. There was a lot of activity in using phage as a realistic alternative to antibiotics. And then, you know, lots of people published lots of papers, you know, companies, you know, did some trials and show that it worked. And then, you know, I don't think the situation became grave enough for companies to really go, okay, we need to look at this as a serious alternative. But I think we might be there now. And so what you've seen over the last 2 or 3 years is more funding going into it, companies seriously looking at it. And I mean, there were issues for companies as well, because if you're going to go down that route, you're going to invest a lot of money. And the regulatory framework wasn't there around phages the way it is around antibiotics. And there was a risk for companies, you know, taking that on. And, you know, if we invest all of this money in looking at these as alternatives, you know, what if we don't get the regulatory approval down the line, and what happens there? And so over the last 2 to 3 years, we're starting to see momentum again there, because, you know, we're at a crucial point, and, you know, the EU and the WHO and everybody's saying this is one of the major global challenges, and we need to find alternatives, and not just find alternatives, but, you know, look at ways of reducing what we're currently using, and use antibiotics in combination with phages. So there's lots of activity going on in this space. And as I mentioned in my talk, I think it's that area that'll set the ball rolling for every other area where phage can be used, because that's the critical one.
Okay. And a follow-up question then, someone has mentioned that yesterday they were talking about TB and beef. Is there any work being done around phages in M. bovis?
Yeah, absolutely. And my colleagues in the Cork Institute of Technology have been working on Mycobacterium for many years with bacteriophages against Mycobacterium tuberculosis, and have done actually some work here with the team at Teagasc. So there is activity going on there. So I suppose one of the attractive things about phages, and you know, if you can find one, you can apply it. You know, it's just finding the application and figuring out how it might work. But certainly there is activity going on in that space as well.
Okay. And we are running out of time, so I'm just going to have one more question for Fiona. If the coronavirus genome has a small genome size and a small number of proteins, does that mean that the probability of a new variant that is resistant to vaccines is less likely than if it was a more complex virus?
Unfortunately, no, because really it is just the spike protein is the major protein that would affect vaccine efficacy. That said, the SARS-CoV-2 has what we call a proofreading enzyme. So if you remember, I said that mutations are introduced during DNA and RNA replication all the time. But actually, because it has this proofreading enzyme, its rate of mutation has been a lot lower than other viruses of this type. So for instance, the influenza virus has a much higher mutation rate. And that's why we have to have those boosters every year. So up until now, the mutation rate has been quite low, and that was good news for us. But then, as John said, the fact is that vaccines will put a selective pressure on the virus to mutate. And we are, I think, seeing that probably already. I think the worst thing that can happen is, and I know there's talk of this happening in some countries, giving one dose of the vaccine and the 2-dose vaccines, giving one dose and then opening up the economy. But with one dose of the vaccine, you only have about 30% immunity, if that. So by opening up the economy at that stage, you're putting a huge selective pressure on the virus to mutate, to make that vaccine completely ineffective. So it just means we all have to just hold tight for another while and get our shots, get our vaccines, get our 2-shot vaccines if necessary, and get as much of the population vaccinated as possible. And I think, you know, at that point we'll be in a very good position.
Okay, I really like John's line that this is evolution in action.
Yeah, yeah. And as I said, all this virus wants to do is replicate. You know, we're just stopping it doing that, and it's just going, okay, you know, well, what do I have to change to let me, you know, go back and infect people again? And in some respects, it's a very sneaky virus because it does it quite well in so many people without symptoms, that it does manage to spread so much.
Okay, thanks, Fiona. And I'd just like to thank the 3 presenters again. I think your 3 talks were absolutely brilliant. I really enjoyed listening to them. I hope everyone who joined us enjoyed your presentations and the Q&A session. We didn't get to all the questions, so we will send them around to the researchers afterwards and they will answer them, and we'll put them up on the website with the PDF version of the slides. Our next webinars in the Research Insights series are around the theme of advances in animal breeding coming from our Animal and Grassland Research Programme. They'll begin in 2 weeks' time on the 3rd of March at the same time. So for now, I'd just like to say thank you very much for joining us today. I know we ran a little bit over time, but I think it was worth it to hear the question and answers. Thank you very much. I hope you'll join us again. For now, it's on.